Hi everyone. I'd like to start by thanking the organizing committee for inviting me to give a plenary talk at this year's EMPSEB conference. I actually attended EMPSEB as a PhD student in 2014 and 2015, and I have very fond memories of that. And I was then on the organizing committee for EMPSEB 2015, so I feel like I've come full circle now by coming back to EMPSEB as an invited speaker. Although I'm now a lecturer at the University of Aberdeen, the work I want to tell you about today is based on my research that I did at University of Glasgow and University of Edinburgh. And I'll start with my postdoctoral research at Glasgow, where I was looking at how behavior and other phenotypic traits might evolve in light of climate change. I think all of us here know that climate change is real and it's already happening. In fact, it's arguably the greatest threat to humanity in the 21st century. Especially relevant for us as biologists are the wide-ranging impacts of climate change on biodiversity. Ectotherms like the organisms shown here are particularly vulnerable because changes in ambient temperature directly influence their body temperature. So there has been a lot of research in recent years trying to understand how ectotherms might adapt to increasing temperatures. For example, we might expect increased temperature to lead to changes in development, morphology, behavior, and physiology with interactions between all of these traits. And these are examples of direct effects of temperature on a focal organism. But there might also be indirect effects of temperature. For example, if an increase in temperature changes the abundance of predators or the abundance of prey, these might then lead to changes in our focal organisms' development, morphology, physiology, or behavior. And changes in those traits can, of course, lead to feedbacks between the focal organism and the predators or prey. So the main point here is that the effects of climate change are very complex and difficult to predict. But being able to predict these effects is critical for management and conservation efforts. What we know so far is mostly based on lab experiments where animals are exposed to high temperatures for only one or a few generations. These studies are, of course, very valuable, but they can only tell us something about short-term adaptation or plastic responses to temperature. We still don't know how animals will adapt to a warm environment after decades or hundreds of years. So this leaves us with the seemingly impossible task of trying to predict the future. Since we unfortunately don't have a time machine at our disposal, we decided to try the next best thing. Which brings us to this powerful natural experiment in Iceland, which provides us with a window into the future. There is a lot of geothermal activity all around Iceland, which means that there are many lakes that are much warmer than normal lakes, which are at ambient temperatures. This small freshwater fish species, the three-spined stickleback, is found in both warm and cold lakes in Iceland. So by comparing fish from adjacent lakes with different temperatures, we can identify the evolutionary changes that take place when fish are exposed to warm environments for decades to hundreds of years. So far, we've identified 10 different pairs of warm and cold populations around Iceland. And our warm sites tend to have a low of about 15 to 20 degrees Celsius in the winter and a high of 20 to 25 degrees in the summer. On the other hand, the cold sites are under ice in the winter, with temperatures just above 0 degrees Celsius, and in the summer, the high is around 10 to 15 degrees. As an example, this is what a typical Icelandic lake looks like in the winter. It's completely frozen, with the ice being over half a meter thick. So to sample fish in the winter, we had to use an ice auger to make a hole in the ice. But we had a much easier time catching fish from the warm lake, shown here on the right, which never freezes. This photo was taken on the same day as the one on the left, and it was taken about 2 kilometers away from the cold lake. In fact, despite the sub-zero air temperature and the snow in the surrounding area, you can see green vegetation along the edge of the bay, which I was really amazed to see in the middle of the winter in Iceland. That was just one example of a warm-cold population pair, but as I said, we've identified many more such lakes all around Iceland. And I think this is a great system for studying the effects of temperature because it provides us with repeated and independent populations that have been exposed to different temperatures for many generations over a small geographic scale. This small geographic scale is important because it avoids the confounding factors that can be a problem when comparing populations from different latitudes or different altitudes. So we took advantage of this natural experiment to look at how living in a warm environment changes behavior, morphology, physiology, and development. This is a list of all the traits that I looked at during my postdoc, which I obviously won't be able to talk about today, but I'd be more than happy to chat about any of this work later. As a quick summary, I found strong divergence in most of these traits when comparing fish from cold and warm lakes. For example, using geometric morphometrics, I found that fish from warm environments tend to be more deep-bodied, they have smaller eyes, and shorter jaws than fish from cold environments. Here, each panel shows one population pair, and on the y-axis we have standard metabolic rate, and on the x-axis we have the temperature that the metabolic rate was measured at. And the general pattern here is that fish from warm environments, shown in red, tend to have a lower metabolic rate than those from the cold environment, shown in blue, when they're measured at the same temperature. But surprisingly, fish from warm environments prefer cooler water when given a choice in the lab. The average preferred temperature was around 13 degrees Celsius for fish from both warm and cold environments. The study I want to really focus on for today looked at how temperature might influence sociality. But first, why should we expect to see any relationship between temperature and social behavior? Well, sociality is quite common and taxonomically widespread, but there is a lot of variation in the extent of this social behavior, not only among species, but also within species. To understand this variation, we need to think about the costs and benefits of being social. For example, animals in groups may benefit from improved predator detection and avoidance. Animals that hunt in groups can also catch much larger prey than they would be able to individually. But of course, sociality also comes with costs, such as a higher risk of disease in animals that live in groups. There might also be more competition for food, nesting sites, or other resources. And these costs and benefits of sociality vary with environmental conditions, including temperature. Variation in temperature can alter the animal's metabolic rate, the availability of food in the environment, the abundance of predators or parasites, and so on. So variation in temperature may drive variation in sociality. To measure sociability in the sticklebacks, we placed a focal individual in the middle compartment of a tank that was divided into three. On one side there was a group of 4 fish and the other side was left empty. And we recorded the behavior of these focal fish for 30 minutes and then analyzed the videos to determine how much time these focal fish spent close to the shoal. This is an example of such a video from a different experiment that was done on minnows. This focal fish indicated in red is very social and is spending a lot of its time very close to the shoal. In contrast, this second fish seems to be asocial and is not very interested in making friends, spending most of its time on the other side of the tank, ignoring the other fish. And here you can see at the end how we plot the overall space use of these two fish over the 30-minute observation, with the social fish staying very close to the shoal and the less social one just swimming all around the tank in the middle arena. In this experiment, we measured sociability at a low and a high temperature indicated by the color of each box, and we used sticklebacks from cold lakes and from warm lakes indicated by the color of the fish. This experimental design allows us to examine both plastic and evolutionary responses in sociability in relation to temperature. And I should also point out that the group of fish that we used for the shoal was always from the same thermal habitat as the focal fish. Here we have water temperature on the x-axis and distance from the shoal on the y-axis. So larger values here indicate that the fish were less social, and smaller values indicate that they were more social. The first result was that at a low temperature, fish from the warm environment were less social than those from the cold environment. And the same was true at a high temperature. There was a slightly smaller difference in sociability between fish from warm and cold environments at this high temperature, but this wasn't significantly different from their behavior at the low temperature. So this tells us that fish from warm environments are less social, but they don't adjust their behavior depending on the water temperature. But what this experiment doesn't tell us is whether these differences in social behavior are genetically determined or environmentally induced. Since we used wild-caught fish, we cannot eliminate the possibility that rearing temperature is what determines sociability later in life. So to answer this question, we did another experiment where we first bred wild sticklebacks from a cold environment or a warm environment and reared their offspring at a low or a high temperature. And we did this again for another generation to minimize parental and other epigenetic effects. We bred these F1 fish and reared their offspring at the same temperature as their parents. So the offspring of these parents were reared at a low temperature, and the offspring of these parents were reared at a high temperature. And our second experiment focused on this F2 fish. If sociability is genetically determined, we would expect that fish whose grandparents came from a cold environment would be more social than those whose grandparents came from a warm environment, regardless of their rearing temperature. On the other hand, if sociability is environmentally induced, fish that were reared at the same temperature should be equally social, regardless of their genetic background. So what did we find? Here we're comparing fish that were reared at a low temperature and whose grandparents came from either a cold or a warm environment. And I was very excited to see that fish whose grandparents came from a warm environment were less social, consistent with the pattern we saw in the wild fish. And we found the same pattern yet again when comparing fish that were reared at a high temperature. Those whose grandparents came from a warm environment were less social than those whose grandparents came from a cold environment. Going back to these possible outcomes, it looks like our results do support this explanation that sociability is genetically determined. However, if we plot these four treatment groups together, we see that rearing temperature also plays a role. When we look at fish from a warm origin, we see that they were less social if they had been reared at 18 degrees Celsius compared to 12 degrees Celsius for 2 generations. And similarly, fish from a cold origin were less social when reared at a higher temperature. So the thermal environment during early development also seems to play a role, even though it doesn't completely erase the heritable differences between fish from different thermal origins. To sum up this first part of my talk, we found that social behavior is heritable but also influenced by rearing temperature. On the other hand, there was no evidence for plasticity in response to temperature changes in adulthood. Our results also suggest that rising temperatures might lead to a reduction in sociability. And this might be driven by a change in the balance between the costs and benefits of being social, but it's still unclear whether this is adaptive, and that's something I want to look into more in the future. For the second part of my talk, I want to switch gears a bit and tell you about some of my work on parental care behavior in a different study system. This next study was inspired by a 2002 paper that looked at the effects of biparental care on offspring fitness. And they found that offspring in nests with only one parent actually performed better than offspring in nests with two parents. This might seem surprising and counterintuitive at first, but it makes more sense if we consider sexual conflict between the two parents during biparental care. So, what is this sexual conflict about? Well, it means that parents try to shirk their duties and shift as much of the workload as possible to their partner. This conflict can lead to arguments about whose turn it is to do the dishes or change the baby's diaper, etc. And if both people argue that it's the other person's turn to wash the dishes, the dirty dishes might sit there for far too long until one of them finally caves in and does them. And in this case, the person that didn't do the dishes might be considered the winner of this evolutionary conflict. Similarly, if you have a crying baby, the desired outcome is a happy sleeping baby. But ideally, you want to get this outcome without doing any of the work, meaning that your partner takes care of it instead. In general terms, evolutionarily speaking, each partner wants to maximize their benefit while minimizing the cost they pay in terms of their time and effort. In non-human animals, this means that the offspring might end up receiving less care from their parents because of sexual conflict, as was the case in the study I mentioned earlier on zebra finches. I found this really interesting and decided to test this question in my study system, which is burying beetles. Burying beetles are great for testing this because they have facultative biparental care. This means that sometimes both parents look after the young, and sometimes the caretaker is only the male or the female. So we can do some interesting experiments comparing how offspring perform with one versus two parents. One thing that makes these beetles really unusual and fascinating is that they bury dead animals and then breed on them, which is how they got their nickname as nature's undertakers. What happens is, once they find dead animal, they remove its fur or feathers, they roll it into a ball, and they bury it into the ground. After burying the carcass, they lay eggs around it in the soil. The larvae hatch a couple of days later, and they start feeding on the carcass. The larvae can feed on the carcass on their own, but the parents also feed them by regurgitating pre-digested carcass into their mouths. And this looks similar to what happens in many birds, such as penguins. The larvae beg to the parents by raising their head and waving their legs or touching the parents. This is a short clip of a female feeding her larvae where you can see some of this begging behavior with this larva here waving its legs and touching the parent. But there is a small problem with using a dead animal as a nursery and food source for your offspring. The bacteria and fungi on the inside and outside of the carcass will make it unusable unless the beetles do something about it. This is a time lapse of a dead mouse that's decomposing naturally over a 10-day period. Bacteria in the gut begin to break down body tissues, releasing gas that accumulates in the intestines. This makes the body expand until it bursts a couple of days later. And now that the carcass is a few days old, you start to see lots of fungal spores. There's lots of fungal spores all over the fur of the mouse, and they start to grow very quickly. This fungus will quickly take over and turn the mouse into a cloud of fungus. Since the breeding cycle of this beetle is around 10 days, the parents need to maintain the carcass in good condition to prevent this from happening. To do this, the parents start by removing the fur, and then they make a hole in the abdomen of the mouse to remove the gut. Removing the gut will prevent the bloating and then deflation that you saw in the video. They also start depositing antimicrobial substances on the skin of the carcass to prevent bacterial and fungal growth. They secrete these substances from their mouth and anus, and they cover the whole carcass with them over and over again throughout the whole breeding period. So as you can see, parental care is quite elaborate in this species, and the parents are clearly important for improving offspring fitness. But does it matter whether there's two parents present or just one parent? To test this, we had three types of families. Here, the male is indicated in blue, and the female is indicated in orange. In our uniparental treatments, we gave each parent one mouse of a standardized size and 15 larvae, whereas in our biparental treatment, we gave parents two mice of a standardized size and 30 larvae. We did this so that the workload for each parent would be the same in the single parent and two parent families. This also standardizes the amount of resources available to the parents and to the offspring. If the effect of the mom and the dad on the offspring is simply additive, we would expect larvae to do equally well with two parents or one parent. Another possibility is that larvae do worse with two parents because of sexual conflict, which leads to a lower amount of care per individual offspring. The third and last possibility is that offspring do better with two parents than a single parent because of task specialization. So what did we find? The first result was that the males spend less time feeding the larvae when they're paired with a female than when they're looking after the offspring on their own. And then females are forced to compensate by working harder when they're paired with a male. And this is evidence for sexual conflict over parental care. This is a short video showing this result in action. The female on the left is getting mobbed by all the hungry larvae, while the male is just standing around grooming himself here. I think this video says it all really, and you can see that towards the end here, the female is actually toppling over because of all the larvae trying to get food from her. Because females in pairs provided extra care to compensate for the lazy males, in the end, the total amount of care received per offspring was the same in the families with one parent versus two parents. Given that total care was the same, we might expect that the offspring would fare equally well with one parent versus two parents. But that's not what we found. Somehow, offspring that were looked after by two parents did better. They had a larger body mass at the time of independence, and they were more likely to survive to adulthood. So there's definitely a benefit to the offspring when both parents are present during development, although the mechanism behind this is still unclear. The take-home message is that it can be beneficial to have both parents present despite sexual conflict, and even though the total amount of care isn't any higher. So the next thing we need to find out is what is causing this effect. What is this magical thing that happens that improves offspring fitness when the two parents are together? To finish up, I wanted to mention briefly what my current research program at Aberdeen is about. Most of my work at the moment is looking at animal behavior in the context of environmental change. The first theme is on how environmental change may alter animal behavior, and the second thing theme is whether such changes in animal behavior can then influence the capacity of populations to adapt to environmental change. Lastly, I just want to give a big thanks to everyone that contributed to lab work and field work, to the funding bodies that supported this work, and last but not least, all the sticklebacks and beetles that gave their life for science. And thank you for your attention.